In this microbes and household products, um, what we are going to discuss is what type of microorganism we are using for what for. We know very well microbes of various types are used for making the curd, for making the fruit juices, for making the dough, for making the butter of various varieties. For that, we are using microorganism, right? If you're talking about the microbes, basically, in the kingdom, whenever you're looking about the bacteria, are having various size and shape. If you're talking about the bacteria is having various size and shape, not only the rod-shaped bacteria, not only spherical-shaped bacteria, all the bacteria of various size and shape we are observing in the bacteria. So we need to understand the bacteria of various size and shape are having various habit in the sense in terms of not only survival, not only multiplication, in terms of having a communication with the other group of microorganisms. We need to understand that basically the beneficial microorganism only we are going to study in this chapter. We are not going to study the harmful microorganism. So microorganism are producing the valuable components which should be purified and then should be sold in a proper preservative way. Why? Because the particular valuable components should not lose the activity. If you are entering into household products, basically many household products are there, not only the curd, not only the dough, many varieties of microbes are there which are mainly responsible for making the high quality products. You know very well, in the deep part of the ocean, at 100 degrees Celsius, there are some microbes are living. So at high temperature, those microbes we used to call as thermophilus bacteria. So we know very well the thermophile is a bacteria. So this thermophile bacteria we used to call as the temperature resistant bacteria. Temperature resistant bacteria, high temperature withstanding bacteria are called thermophile. These bacteria are very, very important for the production of valuable enzyme which is had the credential to digest the protein. We know very well the diverse microbial world. In the diverse microbial world, not only protozoa, bacteria, fungus are existing, many viruses are also existing. So sometimes we are using bacteria, sometimes we are using the fungus, sometimes we are using the viruses for getting the valuable components, right? So in household products applications, we are not using virus. For making the biofertilizers, for making the biopesticides, we are using viruses, right? So now what we need to understand basically in the cultivating the microorganism, when we are cultivating the microorganism, microorganisms are cultivated with the proper nutrition medium. Proper nutrition medium in the sense, for example, I am a specific fungus. I require specific pH. I require specific temperature. I require specific amount of nutrition material. I require specific amount of water quality. I require the specific quality of the the proper aeration. Aeration means in the sense oxygen circulation. So proper aeration, oxygen circulation, water quality, temperature quality, pH quality, and nutrients supplementation is very, very important. So what you need to understand basically the nutrients are very, very important to maintain the beneficial microorganism to survive so that we will get the valuable components, right? So in the microbes, basically, the microbes of various quality in terms of not only in terms of size and shape and also the survival efficiency also we need to think by thinking the pH temperature parameters. In this chapter we are going to discuss about the high quality microbes which is used for the various type of microbial components extraction. So you know very well the curd is making the vitamin B12 that we are going to discuss. Before discussing about the curd in terms of producing the vitamin B12, let me discuss about this bacteriophage. The picture what I shown for you is a harmful microbe. This harmful microbe bacteriophage, whatever we shown here, is used as a vector to introduce a desirable gene into the bacteria. For example, I need to introduce high quality gene into the bacteria. For introducing high quality gene into the bacteria DNA, what we are doing is we are using the bacteriophage. We are using the bacteriophage. So bacteriophage is a virus that infects the bacteria. So the virus which infects the bacteria is nothing but bacteriophage. By using bacteriophage, we are introducing the desirable gene into the bacteria, and we are making the bacteria to reproduce more efficiently with the valuable components, right? Now we are going for the next important concept. The positive role of the microbe we are going to discuss. What is the positive role of the microbe? You know very well some microbes are playing a crucial role in producing the valuable components. That valuable components role production, the valuable components production role is called what? Positive role, like this in this context we are saying, right? So we know very well the microbes after manipulation in terms of gene, we are using the microbe for medicine, for agriculture, and for industry. You know very well the microbes are used to uh, control the pest. Microbes are used to control the worms, not only pest, not only worms, not only insect, not only the other microorganism. We can control other microorganism by using the other microorganism. Like this, we can do one by one in detail, right? So now, what we need to understand basically, the microbes of different quality are used for various type of things, not only for medicine, even for agriculture. You know very well. Uh, Here, the microbes in everyday. Okay, the silent culinary alchemist. Why we chosen this term? Why? Because from microbes we are getting valuable components, not only the processed food material, not only vitamin B12 rich diet, like this mineral rich diet also we can get it. First, let me start discussing about the curd production. By using the lactobacillus bacteria, lactobacillus genus name we already had given. Here, you know very well, the species name is Lactobacillus lacti. This Lactobacillus lacti species is very, very important for making the vitamin B12 in the case of curd. Uh, are you getting my important or not? Bacteria, which bacteria we are using specifically? You know very well. Uh, the Lactobacillus lacti is a bacteria which is having the credential to produce lactic acid by utilizing lactose. So the genus name of the Lactobacillus lacti is this, and then the species name is lacti. That is a message we need to understand. Next basic message what you need to understand is, if you're talking about the milk sugar lactose, the milk sugar lactose is very, very important. Milk sugar lactose. So milk sugar lactose is very, very important to make lactic acid in the curd. The curd lactic acid is formed because of the lactose, which is a milk sugar, by using the bacteria called Lactobacillus lacti. So now we know very well the curd is not only rich in vitamin B12, not only in rich in vitamin K. So vitamin B12 and vitamin K is very, very important for making the production of the hemoglobin. For making hemoglobin, I require vitamin B12. For executing the blood clotting process, I require vitamin K. So this vitamin B12 and vitamin K rich curd is formed from the lactose or the milk by using a bacteria called Lactobacillus lacti, right? So now we used to call simply as LAB. Now, if you're entering into the dough fermentation process, in the dough fermentation process, what you need to understand is, for making the dosa, for making the idli, we require bacteria. When we are mixing the bacteria along the dough, we are getting the dosa and idli, and this dosa and idli are produced with bubbles because of the carbon dioxide liberation. The carbon dioxide is, once it is liberated, what is exactly happening in the doughs and the bread? You are getting a lot of holes in it. In the bread, a lot of holes are there. In the case of doughs, a lot of holes are there. That is because of the carbon dioxide production. You know very well the bread dough is formed with the help of the baker yeast. The baker yeast is also called as Saccharomyces cerevisiae. Saccharomyces cerevisiae is a yeast name. All the yeast are not used for making the bread, only certain yeast are used. We know very well the Saccharomyces cerevisiae. In this, one is genus name, another is a species name. One is a genus name, another is a species name. So the genus name is Saccharomyces and the species name is Cerevisiae. So this bacon is Saccharomyces cerevisiae is used for making the bread, right, of different quality. Now, if you're talking about the next important message in making the bread, carbon dioxide liberation by fungus yeast is required. So fungus, all the fungus can't make the bread, only certain fungus and the two yeast and the two not all the yeast. I told Saccharomyces cerevisiae is used for making the bread. Now, if you're talking about the next important component is cheese making process. In the cheese making process, uh, here one thing is um, flavor, the cheese flavor and characteristics of the cheese is because of the various microorganism application. You know very well the Propionibacterium chardonnay. Again, back I'm pronouncing it. So Propionibacterium chardonnay is a bacteria which is used for making the cheese, which produces huge concentration of what? Carbon dioxide. So this Propionibacterium chardonnay is a bacteria which is used for making the cheese. And in this cheese, the carbon dioxide is produced that you can see in the right hand side. Whereas the Roquefort cheese is formed with the help of the fungus. The Roquefort cheese is formed with the help of the fungus, which also produces a big hole in the cheese. We know very well in the particular cheese making industry why we are using the microorganism. Why? Because it is used for making the cheese with a lot of vitamins. Why? Because the cheese rich in vitamins are very very important for supporting the various cell activity. You know very well various cell activity in the sense what the cell activity in terms of cell multiplication, in terms of cell division, in terms of cell growth. uh, you know very well cell growth, cell division, cell multiplication, all these things are executed with the help of vitamin. These vitamins are of many varieties, not only fat soluble vitamin, not only water soluble vitamin. So vitamins of water soluble vitamin category, fat soluble vitamin. So one is water soluble vitamin, another is what? Fat soluble vitamin. So one is water soluble vitamin, another is fat soluble vitamin. That is very very important for making the high quality diet which satisfies all the human need for doing the all metabolic activity. So we required water soluble vitamin and fat soluble vitamin that is rich in cheese, which is made either with the bacteria or either with the fungus. If you're talking about the fermented beverages, before entering into the fermented beverages, we will enter into the examples in the curd formation process in the exam. Basically, if you're talking about, they will ask how the curd formation is happening. So if they're asking the question, we need to write a lot of paragraphs, subparagraphs, and then we need to write with a lot of points. So in the case of board exam, when you are writing this curd formation process, uh, here Lactobacillus lactis is a bacteria which is used for making the vitamin B12. Like this, we already told you. In this, the milk protein casein is coagulated, forming a gel-like structure. Forming a gel-like structure. So this protein we used to call as whey protein. We know very well. The whey protein is having high value in giving the protein supplementation, right? The curd is rich in protein as well as in lactic acid. Here you know very well, but nutritionally enhanced. The curd is having the vitamin B12. Not only vitamin B12, vitamin K is also rich in the curd. But vitamin K is less, vitamin B12 is maximum. Why we require vitamin B12? We know very well for RBC formation or red blood cell formation, we require vitamin B12. The chemical name of the vitamin B12 is cyanocobalamin. Cyanocobalamin is a vitamin B12 chemical term which is used for making the RBC so that the breathing process will be executed more appropriately. So gas exchange, you know very well, gas exchange. Okay, gas exchange or breathing process is executed more nicely with the help of the RBC and hemoglobin formation, right? So now, if you're talking about the lactobacillus lacti, this is the way your answer presentation should supposed to be. Now we are coming for the bread formation. In the bread formation, basically, uh, how the bread is formed. Um, in the case of bread, you know very well, the carbon dioxide production in the bread is mainly because of the Saccharomyces cerevisiae. The yeast Saccharomyces cerevisiae is also used for ethanol production. You know very well the ethanol of different quality. Ethanol of different quality, in the sense, uh, here you know very well, not only here in the CBSE textbook they already given the whiskey, brandy name, beer, rum, gin. All these components name they already had given in the case of CBSE biology textbook. So based on that, we are discussing all these components in detail. You know very well, some are obtained with fractional distillation, some are obtained without fractional distillation. Fractional distillation means in the lower grade, we already had studied separation of the molecule based on the differential boiling point. Separation of the molecule based on the differential boiling point is nothing but what? Fractional distillation. So here by using fractional distillation, we are making different type of beverages from the fermented products. We know very well the yeast is used for making the beverages. That beverages, which is nothing but ethanol, we should not allow methanol production for specifically for the food industry. So why? Because the quality check is very, very important. Methanol, if it is produced by the fermentation exuding yeast, then what will happen is the kidney failure, lungs failure, liver failure will happen. That is the reason why appropriate checking is very, very important. And second thing is not only for making the bread, not only for making the ethanol, we are using yeast for cloning process also. That we will be studying in the biotechnology chapter, which is going to come after 2-3 session of Go to University team. Right now, if you're talking about the bread, how it is formed with the help of the yeast, we already told you. You basically we used to mix the yeast along with the bread, along the dough, and then this yeast produces a lot of bubbles in it, and then that bubbles we are seeing in the case of bread. Okay, we are seeing in the case of dough also, right? So now what we need to understand basically, um, the ethanol production. Ethanol production is done with the help of the yeast Saccharomyces cerevisiae that we already told you, right? Now we are coming for the next phase of explanation is, you know very well, the curd formation and bread formation, how we narrated in terms of many subheadings. Under the each and every subheadings, we give a lot of many points. Those should be narrated in this way for getting the better score, right? Now we are coming for the next major explanation. After the this is called what worked examples. Also we can say hot questions. Now we are going for tips for the exam. In the tips for the exam, so all the numericals like this we are not saying as like max. In the case of biology, terms we need to remember not only the microorganism name, not only the process name. For example, let us consider fermentation we use now, right? And then we used about the lactobacillus lacti. So fermentation word, lactobacillus lacti, all these are all the various type of terms we are using more frequently in this chapter, right? So we know very well in this chapter for fermentation we are using Saccharomyces cerevisiae yeast. Okay, we are using lactobacillus lacti for making the curd. So microorganism for which process we need to remember. Second concept is what is the outcome of this particular fermentation process. Fermentation outcome, ethanol production. Fermentation outcome, bread production. Fermentation outcome, dough production. So these are the things we need to understand when we are talking about outcome. So whenever you are going for the exam preparation, for each and every concept, what is the process name? What is the microorganism name? What is the outcome of this reaction? And what type of help the particular component, microbial component, is going to help us. Like this, we need to keep in our mind so that facing the exam is very easy, right?